welcome back to the lecture series in physiology. So, we finished the second lecture of the 5th week, and by this time the whole process of action potential, or at least the way the streaming of positive charges are taking place, is kind of clear to you. So, now today what we will do, we will assign the ions which are fluxing in and fluxing out in what sequence. So, discovery of the process of action potential dates back to the earlier part of the 19th century, or even at the fag end of 18th century, where these observations were made by some absolutely patient and extraordinary league of physiologists. But in order to explain this kind of sudden changes of membrane potential in specifically certain kind of cells, and our real understanding, or the current understanding, I must say, real understanding, because we keep on understanding these processes. It took us almost 100 years. It was during 1940s, 30s for the first time, especially just before the Second World War, and kind of progress got delayed because of the war. It was the credit for first time formalizing it. It was known, but really to formalizing it mathematically goes to Hodgkin and Huxley. They were the very first one who kind of developed a mathematical model based on the findings of the action potentials, and I could see the action potential on a big nerves, sea slug, aplysia, because they have a really big axon. They could really probe the electrode, they could repeat their experiments, and they came with a mathematical model of the observations what they made and what we have already drawn in previous two classes, the exact nature of the action potential, where from minus 80 millivolt all of a sudden it shoots up, goes above 0. So, it means from a polarized state the cell goes to a depolarized state, and it goes to positively polarized state because it overshoots the 0, and it again comes back to its polarized state, but before really merging with its polarized state, negatively polarized state at minus 80, it hyperpolarizes negatively further, and then it comes back. And we have also discussed in the previous class the slope of falling down of post positive polarization varies from cell type to cell type, and that slope is kind of a characteristic of the dynamics or the motion of different kind of ions which are taking place. So, before I put it down in the presentation sheet, let me give you verbally what is happening, what we know in last 100 years. The first thing what happens is, because of some impulse which a cell receives, a neuron receives, some of the positively charged gates. So, our cells consist of lot of gates. Some gates, before let me tell you the gates. Some gates open outside the cell, some gates open inside the cell, and most of these gates behave like diodes. In other words, when we talk about diodes, we talk about current flowing in one direction. So, these diodes, what we are bringing the analogy of diodes, these gates either will allow something from inside the cell to go out, or will allow something from outside to go in. So, these gates are called, could be mostly they are voltage gated or ligand gated. I have introduced two more terms. So, let me just start putting them down. So, today we are into week 5, lecture 3. So, what I told you is, if this is the cell, then you have these gates or gates like this, which will you allow. You have pumps sitting out there which could pump a specific kind of ions outside and pull in another kind of ions inside. And most of these gates are called ion channels or ionic gates, you can call them, for your better understanding. And most of these ion channels opening and closing is not random. Okay? Opening and closing is a function of opening and closing is a function of voltage across the membrane, or sometimes they are governed by some ligands. What does these two words means? These two words means at a specific voltage, at a specific voltage. So, say for example, so we know with respect to inside, outside is more positive. So, you can represent it as a battery, right. So, this is the negative and this is how the membrane looks like. So, if you measure the voltage across it, which we know is sitting at minus 80 millivolt, sometime minus 70, minus 40 to minus 80 millivolt. Now, if this voltage across it changes, then this change across it could lead to the opening or closing of some of these ion channels. That is why we call them, the word turn out to be voltage gated ion channels. Okay. Similarly, at times there are some ligands. Ligand means something which comes and binds to the cell, binds on the surface of the cell. Upon binding of certain things, these channel gets open or close. So, say for example, suppose here you have a channel like this, which is supposed to allow the ionic flux to move from outside to inside, and this is your outside the cell and this is inside the cell. Something is supposed, so say for example, so it in close proximity there will be some ligand gatings. So, say for example, this is where the ligand or something will come and bind. This is that molecule. So, as soon as this molecule comes and binds to this site, this channel open, and these kind of channels which are function of binding of some XYZ ligands, and this ligand could be anything and everything. It could be a photon, even it could be a photon, it could be some chemical. Okay. So, these kind of channels are called ligand gated, because it is the ligand binding which leads to the opening and closing of the channel. Okay. Ligand gated ion channels and voltage gated ion channels. So, when action potential is generated, what possibly happens in the beginning, a ligand possibly comes. I am talking about the sensory structures, will come later into the central nervous system. There we will talk, there are a few other modifications which takes place. certain ligand comes and binds and it opens up certain inward cation channels. What does that mean? That means cations which are positively charged, right, and ions which are negatively charged. Now, suppose I am imagine myself as the whole single cell. Now, it is a ligand comes and bind here. As soon as the ligand comes and bind here, it opens up certain channels which will open or which will allow from outside something to get inside the cell, and it will only allow the positive charges to get inside the cell. So, upon ligand binding, if the positively charges are entering inside the cell, what will happen? Your cell is sitting at minus 80 millivolt, right. So, automatically your membrane voltage is going to become slightly more positive. Maybe from minus 80, it may go up to say minus 75 or minus 70 or maybe minus 60 or maybe minus 30 or 40. Now, here is the catch. If because of the binding of a ligand or some other means, inside across the cell becomes positive up to a threshold point, and that threshold varies, it's around minus 40 or minus 30. Okay. If it reaches all the way up to minus 40, then that leads to the opening of all other cation channels, and the biggest number of cation channels which allows, let me clarify it, the cation channels which allows ions to move from outside to inside, and the large number of cation channels or the maximum cation channels are the sodium channels. In other words, that will allow the sodium flux to get inside the cell. So, in order to achieve this, so what is happening, what all I told you now. So, here is the cell sitting at minus 80 millivolt. Okay. Let me okay. This is the 0, this is the 0. So, cell is here, and this is telling you millivolt and this is giving you the time in millisecond. So, cell is sitting at say this is minus 20, minus 40, minus 60, minus 80. Cell is sitting here. Now, an impulse comes, some ligand binds or something happens, which opens up cation channels, positive charges started fluxing in. If the positive charge is fluxing in, what will happen? The membrane potential will shift like this, right, agreeing with me, because inside it is minus 80, now you are getting positive charges coming in. So, automatically this will change it. But say for example, this is minus 60, minus 40, suppose it all the way reaches something here, then something amazing happens at this stage. All other channels, all other sodium channels which only allows the sodium ion from outside to get inside opens up in a cascade. It is almost like a wave or a cascade which happens. And then what you see, there is an overshoot at this point like this, because the inside become too positive. There are too many positives which comes in. So, that too many positive lets it to the overshooting inside. So, the first event what you see is, what you have to mark is this line which I am now putting somewhere around here. This is that threshold level. If the threshold level is not reached, action potential will not be shoot. So, you could have change, you could see something like this, it will change and it will fall back, it will change, it will fall back. But if it reaches this threshold, suppose out here it reaches this threshold, then there is no stopping, it will shoot up. So, this is that threshold point where there is something called all or none theory comes into play. Either it will happen, all of them will open up, or none of them will open up unless you reach the threshold. So, there is a threshold for an action potential, and if it reaches the threshold, this leads to something called all, and if it does not reaches the threshold, then it is none. If it reaches and if it does not reaches. If it does not reaches the threshold, it is none. If it reaches the threshold, it will be all. All these positives will open. So, once all these positives will open, inside it will be all positive now. Now, there are two kind of positive charges inside. You have a huge flux of sodium and you have a huge flux of potassium inside. And now, what happens? There are another set of cation channels. Now, I am representing them by pink, which allows a specific kind of cations or positively charged ions to only to move outside the cell. Okay. Those are potassium channels, voltage gated potassium channels. So, what happens now? There are too many positive charges inside. So, there are positive repulsion which happens between the charges. There is a mutual repulsion, there are too many positives. That further, this inside that going all the way to a positive level opens up the voltage gated potassium channels. So, based on the motion or direction of the movement, based on the vector of the ionic flux, there are names to this. So, the first name for the sodium channels, voltage gated sodium channel, is fast activating inward sodium current, which is governed by V stand for voltage gated, Na for sodium channel. Okay. Fast activating invert. Why it is called fast activating? Because if you follow this line, see the rise is very fast, it is really fast, like zoop it moves in. Okay. Contrary to it, if you see the slope of its coming down, it is much more slower, and that coming part of that coming down is because of this outflux, these outflux of potassium ions. Okay. This outflux of potassium ions is called slow potassium current. It is an outward current. Why it is called current? Because outward, because the flux of ions. So, electricity could be defined by two ways, either it will be a flux of electrons or flux of ions. Okay. So, the flux of electrons constitute the solid state electronics, whereas flux of ions constitute the ionic electronics. And based on that, there are whole sets of devices, electrochemical cell, which is an ionic device, whereas a semiconductor, it is a solid state electronic device. Fine. All your batteries, these are all ionic devices, these are all ionic phenomena, which is wet phenomena, because you always need a medium for these ions to travel. These are bulky molecule, bulky structures which has to travel. Okay. So, this is called slow activating potassium current, potassium outward current, okay, governed by these potassium channels. Okay. So, now once the potassium goes out, sodium gets in. Now, of course, the membrane can comes back to its original state, but a cell's homeostasis is different. It stays at a very low sodium concentration. Then comes into play a third player into the game, and that player is a pump. A pump which is, now I will introduce the pump here. These pumps have a very unique way of throwing sodium from inside and pulling potassium from outside. These pumps are called sodium potassium ATPase pump. This ATPase is basically the ATP needed there, where the ATP is being utilized. They are energy dependent process. So, first, if I had to summarize, first a cell has to reach its threshold, which is, I am now marking it in blue, threshold. Once it reaches its threshold and suppose action potential is targeted, what you observe is this threshold has to be because of some cation movement inside the cell. Then the whole family of sodium channels will open and you will be recording a very sharp sodium current here, fast activating inward sodium current governed by voltage gated sodium channels, right. After this, followed by this, inside there will be too many positive charges, right. This too many positive charges will be colliding with each other. There will be huge amount of repulsion. At that stage of membrane potential, the potassium channels which are present there, which only allows ions from inside to move outside. So, they open outward. So, any potassium will be moving out, they opens up, and once they open up, the potassium ion goes out, right. Potassium ion goes out. Now, the inside the cell you have higher concentration of sodium and lower concentration of potassium. Now, the cell has to restore its ionic homeostasis. Voltage homeostasis is not a problem, ionic homeostasis is now in a question. In order to restore it ionic homeostasis, a third set of molecules sitting on the membrane surface comes into play, which is called sodium potassium ATPase pumps. The sodium potassium ATPase pump, their role is, it is an energy dependent ATP dependent pump which takes sodium, it will take 3 ions of sodium from inside and it will throw it outside. Okay. So, there are different configuration and it will take another few potassium from other side, from outside, and will throw it inside. It is something like this, a motion happening like this. Okay. So, at one point of time it throws away more sodium as with respect to its house, because it has to throw away a huge amount of sodium outside. So, there are series of such pumps which comes into play like this. Okay. So, because of this the homeostasis is maintained. So, within this fraction of a moment, what you see an action potential out here, a single action potential, all these things are happening. So, all these things are happening in some microseconds, milliseconds domain, micro to milliseconds domain, all these things are happening. That is why we consider a cell as one small machine which is doing all these things. So, now what happens whenever such an action potential is generated? So, within the cell it propagates like this. So, now you have to realize this action potential, say for example, I talk about an action potential. Let us take a typical neuron. Okay. Now, most of the action potentials are generated. I said most because there are exceptions here and there, are generated here. It is a local event. Mark my word, it is a local event. These action potentials travel because this disturbances travel like this. It is not the whole cell at one point becomes like this. So, they could travel like this, they could even travel like this, reverse direction. They could travel like this, they could travel like this, and they can even travel like this. But these traveling does not happen significantly. They do travel certain degree on these directions towards the dendritic trees, but they are not significant. Why they are not significant? Because your higher concentration of sodium channels are sitting on these locations at the axon hillock. If you remember when I showed you the anatomy, axon hillock. So, that is why the predominant motion of this disturbance is like this. So, here, well, I am almost about to close this class. Let me tell you, direction of action potential is direction of action potential propagation is along the axon, but mind it does not rule out that it can't travel in the reverse direction, but that travel is not really significant. And one more thing, when I told you its action potential is a local phenomena. So, what happens? Suppose something comes and binds here. So, this part becomes all of a sudden become positive. Okay. Once this become positive, there is this electrical impulse gets generated. Then this makes this part positive, then this makes this part positive, then this makes this part positive, then this makes this part positive. Likewise, there is a wave front, just like suppose you throw a stone in the river, in a pond, you see a wave front spreads like this outward. That is precisely what happens in an action potential. Then this part get activated, then this part, then this part, then this part. But if it starts with that all or none, then this will spread like this, and then something happens at this end, which will come in the next class, what happens there. So, remember, generation of action potential is a local event. It does not happen all across the whole cell. It does not happen one point of time. At a particular point of time, it starts at some point. It could be here, it could be here, it could be here, it could be here, it could be here. But then it travels. Then this disturbance spread out like this, just like happens when you throw a stone in a pond. It spreads, like you see the wave fronts. It is just like a wave front, it spreads out. And so once it spreads out, so say for example, if this is, say for example, your axon. So, disturbances originate here, this part become positive, fine. Then this part get restored, then this part become positive, then this part become positive, then this part become positive, then this. So, there is a wave which is moving like this, right. So, you see there is a wave front moving like this, and that is precisely what happens in an action potential. It is not the whole thing become positive in one shot, otherwise the cell will pretty much will die out, because it does not have that much energy to withstand it. It is at spot by spot, it moves, and this whole thing happens in some microseconds, milliseconds. This is all a millisecond process. Okay. So, at every location such things happens. So, now after giving you this brief background about action potential, and this whole process can be mathematically explained, this change, if you read through Hodgkin-Huxley's work. And always one more thing I wanted to project, Hodgkin-Huxley did not know the existence of ion channels. They just predicted there is a possibility of these kind of ion channels. With this, I will close in. We will have more stories in the next class. The story of action potential, one of the most fascinating stories of last century, where we come to know about lot of things about neurobiology, and we will continue with next will be the neurotransmitter and a little bit more about Hodgkin-Huxley. Thank you.